Hello, everyone, and welcome to today's webinar, COVID-19 Pandemic, Emerging Testing Solutions. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thorson. To learn more, please visit thorson.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of the questions as we have time for at the end of the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I would now like to welcome our speaker, Dr. David Engman, Chairman Emeritus of Pathology, Cedars Sinai Medical Center, retired Professor of Pathology, Northwestern University. For a complete biography on Dr. Engman, please click on the Presenter tab at the top left of your screen. Welcome, Dr. Engman. You may now begin your presentation.
Thank you for the introduction. I'm David Engman. I'm the Medallion Chairman of Pathology Emeritus at Cedars Sinai Medical Center and retired Professor of Pathology at Northwestern University Feinberg School of Medicine. I have the following disclosures. An overview of the presentation. I've divided this into three parts. The first will be an update on the COVID-19 pandemic, with a review of where we stand with global infections, deaths, and vaccinations, and two sections on variants and vaccines. The next section, SARS-CoV-2 testing, will review the types of testing and their uses, their pros and cons, and then a section on the challenges for laboratories and healthcare providers with respect to SARS-CoV-2 diagnostics. Finally, I'll talk about COVID-19 in 2022 with an outlook for the pandemic and a discussion of emerging testing trends. First, an update on the pandemic. So global infections, deaths, and vaccinations, most of the audience is very familiar with how these have tracked since the beginning of the pandemic a few years ago, but I'd like to review them nonetheless. First of all, the biology and transmission. Remember that one viral particle, shown on the left, can produce thousands of progeny virus particles from a single cellular infection cycle. The virus is transmitted through airborne droplets primarily. In addition to budding out from the infected cell, as seen with the yellow pseudo-colored virus particles in the right image, the infected cell can also lyse, releasing not just virions, but also building blocks of viruses, including nucleocapsid protein, all the other proteins that make up the virus inside and out. And that will be important later on. In terms of the immune response, we normally control this virus, most immunocompetent people. And the immune response induced by viral infection involves both humoral or antibody immunity on the left side of the panel and T-cell or cellular immunity on the right side. Humoral immunity involves the stimulation of B lymphocytes to differentiate, this is on the left side of the panel, into antibody-producing cells called plasma cells. On the right side, cell-mediated immunity involves the maturation of CD8 positive T cells that then differentiate to become cytotoxic T cells on the bottom right. So the two effector molecules of the immune system are antibodies and cytotoxic T cells, and these are what help us to fight the virus. Antibodies by blocking viral binding to host cell surface, and cytotoxic T cells by killing infected cells. And here is an image of antibodies binding to SARS-CoV-2 virus particles and also in the with the surrounding cells as well. In terms of clinical outcomes, based on Omicron infections, the percent of people with no symptoms is very high. It's 4 out of 5 or even higher. Only a small number of people, 20 to 30%, develop symptoms that usually they can manage at home. No hospitalization required. A very small number of people, maybe 1% with Omicron, require hospitalization, and only 1/10 of those die of this infection. And finally, this syndrome called long COVID is a multi-organ infection, infectious sequela of infection, that involves multiple organs and can affect many people. It's not exactly known, depending on the study, it can be 10% up to 30, 40%, have long-lasting symptoms of this infection. And that's going to be a major challenge going forward, is how to manage long COVID. Over time, I've shown on this chart from the World Health Organization, the number of cases on the left y-axis, and those are in the colored bars that track along the bottom of the graph, and the number of deaths, which is the blue line that runs above in the center of this graph. You can see over time that different continents have experienced the majority of cases at different times during this pandemic. And there's been a very large increase in total number of cases at the very right due to this Omicron variant. I'll talk more about this later. What I've pointed out with the red text is that Omicron has a very low ratio of deaths to cases. That is to say that while the number of deaths before Omicron tracked in the tens of thousands of deaths per week, that is also true currently, but the number of cases has risen from a few million cases per week through most of the pandemic up to now over 20 million cases about a month ago. And so it's no more deadly than it was, but there are many more cases. And that's going to be an important point as we discuss Omicron. This is equally seen here if you look at the table on the right side and with the orange boxes. The cumulative number of cases and deaths gives a case fatality ratio of a little over 1%, 1.39%. That's the number of deaths divided by all the cases in the world up to now. But only in the last 7 days, the case fatality rate is only 0.53%. And that's because Omicron is not as deadly. This sort of reproduces the graph that I showed on the previous slide. And all the details will be available for your review after this webinar are shown in the table to the left. And so I would also point out that the case fatality rate is different from the infection fatality rate, which is an estimated number of the total number of actual infections. And so the infection fatality rate will be lower because there are many unreported infections. And I'll talk more about this as well. A tiny percent of people are actually accounted for by testing. If you look on this graph, which tracks the percent of estimated COVID-19 infections that are actually detected, it's less than 10% in the United States at the present time of actual infections are reported. To be reported, an infection has to be documented, typically by PCR testing. And the reason for this discrepancy between actual and reported include asymptomatic infections are not detected, let alone reported. People are unaware of being infected. Symptomatic infections that are either managed at home with or without self-testing are also not reported. So that gives the tremendous undercount of actual infections. Now let's talk about the variants. What are they and how do they emerge? The SARS-CoV-2 genome is shown at the top. The virus uses a special DNA polymerase to replicate its genome called reverse transcriptase. That reverse transcriptase makes a lot of errors compared to our own DNA polymerase that replicates our own genome and introduces point changes or mutations into the genome. Those mutations can occur in any viral gene, not just the spike protein gene that we're most familiar with when people talk about mutations that might affect antibody binding or might increase the receptor affinity with host cell receptors and promote viral entry. But they can occur anywhere in the genome. Some of those mutations confer the virus with a greater ability to spread, and there are several mechanisms of this I'll describe in a minute. But such a virus can spread better than the parent virus. And different viruses with different pathogenic potential and unique sequences are called variants. Here are the spike proteins of some recent SARS-CoV-2 variants. In yellow is the alpha variant that emerged first in the United Kingdom. Next, we have the beta from South Africa, then the epsilon from California, gamma from Brazil, and delta from India. And I'm only pointing this out to show that each one of these variant spike proteins has a different set of mutations that are colored in the pictures of the spike protein shown above each of the labels. So that's just one example. But remember, each one of these variants may have other mutations that also confer a growth advantage that allowed each one of these to emerge in turn. You can actually see this even better by using the GISAID depiction of variant spread over time. On the left, we're talking about the beginning of the pandemic all the way across the right to the present time. And you can see that at the bottom, the first one was alpha, shown with the letter A there. It's actually Greek capital alpha. Then the beta came on top in the middle of the section, then gamma, then delta, and finally it's not all the ones in between delta and omicron are not shown. Those are the Greek letters, but omicron at the end. So what's interesting here is that each one of the variants arose not from a previous variant, but from elsewhere. So you can see the beta popped up before gamma, which arose a little bit later from the alpha lineage. Delta emerged from beta, and omicron emerged from some part of gamma. So it's very interesting to note that the new variants that were dominating the globe actually arose not from the previous variant that dominated the population. And you can see out on the left, on the far right side, omicron, if you go vertically from those last, medium blue dots there upwards, you'll see that there are no other variants other than omicron at the present time. The only things that are in medium blue are omicron at this point. This in the United States is also depicted this way. And you can see the alpha, gamma, and beta, and the delta is the big orange one. And that came up very quickly to dominate the population back last summer. And now in the late 2021, and now we see omicron in purple has sort of taken over delta. It replaced it very quickly within a month. All the delta was gone, omicron replaced it. The good news is that the infection fatality ratio of omicron is very low, less than 0.2% throughout the entire country, with Vermont having a little bit more. But this is not a very deadly viral variant. Of course, the problem is because we have so many infections, we still have significant death throughout the world and in the United States because the numbers of infections are much higher. How does a viral variant rise in prevalence to dominate in a population? There are many possible theoretical reasons, and some of these are actual. Replication rate, if a virus replicates even a little faster, it will dominate a population fairly quickly. Same thing about the ability to infect cells. The amount of virus produced by an infected cell. Residence in the upper versus lower airway. Viral variants that seem to occupy upper airway, like Omicron, tend to produce more shed virus than those that occupy the lower airway. Concentration in airborne droplets and the persistence of those droplets in the air. Those are other variables that mutations can affect. Longer time of viral shedding. If a person's asymptomatic and actually shedding virus for a week instead of 4 days, or 2 weeks instead of 1 week, that can also promote that viral expansion. The ability to cause asymptomatic infection, where a person's unaware of being infected, may not take the kinds of precautions that he or she might if the person had symptoms, is also can promote shedding and expansion. The ability to evade host immunity. There's been a lot in the papers about this, although there's been a focus on that without necessarily considering all these other mechanisms that can promote variant expansion. And I would note, though, that increased transmission does not equal the ability to cause severe disease, as we've seen with Omicron, which causes less severe disease. So the two are definitely distinguishable, although some of the mechanisms involved in causing severe disease may be listed contained in the list above. This is just one example of, this is actually, I made this myself, just taking two viral variants, theoretical variants, one of which has a 1% growth advantage over the other. And you can see it only takes about 300 cycles before the variant that can grow a tiny bit better, 1%, will dominate that population. So if you were to take a person and infect them with equal amounts of Omicron and any other variant, within a matter of days, the only thing remaining in that person would be Omicron, maybe even less than a few days. Vaccines. What do they do and what don't they do? Well, all 5 major SARS-CoV-2 vaccines that are used in the United States and Canada or Canada work well. You've got mRNA vaccines from Pfizer and Moderna on top, the viral vector vaccines from Johnson & Johnson and AstraZeneca in the middle, and the protein subunit vaccines, Novavax, on the bottom. Throughout the world, you've got lots of different viruses that are in different states of approval, emergency use, etc., in different countries. And all of the vaccines work to a greater or lesser degree. They all induce humoral and cellular immunity. Vaccination reduces symptoms, need for hospitalization, need for intensive care, and death from COVID-19. And infection recovery does an even better job of reducing these. That is to say that if someone is infected and recovers from COVID-19, they actually have slightly better immunity than those who are vaccinated. And of course, many people have been infected and vaccinated. Vaccination and infection recovery do not completely prevent a person from being infected or from infecting others. So while they reduce all those other parameters, a person can still become infected and shed virus to infect others, even if they're vaccinated. And this is also responsible for a lot of silent infection. Silent infection among vaccinated people allows this virus to persist in the population, and I'll focus more on this later. Vaccine efficacy wanes over time, though, as measured by antibody concentrations, typically, but protection from serious illness and death lasts longer than protection from becoming infected. So that's important. People can become reinfected, but often aren't going to develop serious illness that they might have if they were infected primarily before vaccination. What about the boosters? Well, there's a lot of discussion about people now seeking a 4th booster shot and maybe planning on a 5th one later this year. And I should just emphasize that protection from hospitalization and death is longer lasting than the antibody levels might indicate. In fact, people thought that we could just measure antibody levels, and then when they drop to a certain level, you might need a booster at that point. But there isn't a hard, fast rule on what that level is that and so that has not been implemented as a public health policy in any country. Testing. Let's talk about the types of SARS-CoV-2 testing and their uses. The molecules for the diagnosis of SARS-CoV-2 infection and immunity are shown on this slide. In the left panel on the top, we have RNA, the nucleic acid genome of the COVID-19 virus. That RNA is coded by nucleocapsid protein. Those two molecules are internal to the virus. On the lipid bilayer that constitutes the viral membrane, you can find membrane protein and envelope protein. These are two integral membrane proteins, shown in purple and blue. And then at the very bottom, emerging from that surface, is the spike protein, S. It has a couple subunits, S2 and S1, and that's what binds via the receptor binding domain, RBD, to the host cell receptor ACE2 on the host cell. Infection with this virus induces, on the right panel, antibodies that are produced against typically spike protein, but also all of the other proteins that are mentioned, as well as others not shown in the diagram. That is to say that infection with this virus induces a polyclonal polyspecific antibody and T cell response to lots and lots of viral proteins, not just the ones that are in the news. And those antibodies can be used for diagnosis. Let's start out, though, from the top, RNA. So RNA can be used for molecular diagnosis. Most of the RT-PCR, reverse transcription PCR assays, include several genomic targets to really make the specificity very good. This is a very sensitive test. Protein, typically nucleocapsid protein is the most commonly used, can be used for antigen testing. And the presence of antigen or the RNA may indicate infection. And finally, antibodies on the right are used for serology, with anti-spike and anti-nucleocapsid proteins being the most popular assays. Now, the tests for different molecules will be positive at different times post-infection. So shown on this graph, we're tracking the RNA levels in red, antigen levels in orange, the IgM, the early immunoglobulin antibody in blue, and the long-lasting IgG in green. So the RNA test can be positive for the first several weeks, but many people can continue to have detectable RNA in a nasal and nasopharyngeal sample after virus has been cleared. The antigen test is positive only when viral particles are around. So when virus is cleared after several weeks, the antigen test may become negative. The IgM antibody precedes the IgG by a number of days, but typically wanes and then may be absent after a few months, while IgG persists for a long time. This is during and after infection. So depending on when you measure, you'll find positive or negative test results for these markers. Here are the pros and cons of these tests. For the detection of RNA by RT-PCR, one of the pros is that the RNA can be easily detected by this molecular test. That level can be measured by quantitative PCR, and the results are often given in what are called cycle thresholds, or CT. With the larger the CT, the more rounds of amplification were required before the RNA signal was detected. So high CT means lower RNA to begin with, lower RNA in the sample. Low CT means it amplifies earlier, higher RNA, and higher amount of virus. And this can be seen in this diagram with the high specificity of this assay shown on the top panel, the process of RNA amplification in the middle panel by PCR, and the viral amplification curves in the bottom panel, with the high virus on the left, with low CT, the low virus on the right, with high CT. The CT is on the x-axis of that bottom graph. So this is how that's depicted. Now, the molecular testing is the gold standard and is in widespread use throughout the world. It's the most common test up to now. And I'll talk about future testing as we go forward. The cons, it's more expensive. And as I mentioned a few slides ago, viral RNA may be present without infectious virus. Now, RT-PCR may be positive long after the person has stopped shedding virus. And this graph shown here demonstrates that, where there's this post-infectious period where a person can be positive by PCR, shown after that infectious stage that's highlighted in the orange shading there. And that is the period where the person may not be infectious, but may be positive by PCR. And that will be important in the incoming slides. So what about, so the other thing I would mention is that the results are not immediately available with detection of RNA, and this typically requires testing in a laboratory. There are a few assays out there that have emergency use authorization that are sort of disposable thermocyclers, but they're not in very high use at the moment, and they're not inexpensive either. Detection of antigen, well, the pro of this is that a variety of antigens can be used. Nucleocapsid protein happens to be the most commonly used one. And you can have rapid antigen tests that can give results in 15 minutes. And the more high throughput, slightly better laboratory antigen tests can give a result in about an hour. The rapid tests can be done at home. They're qualitative. Laboratory antigen tests are typically qualitative, but can also give quantitative results that may be of use. Antigen tests are less expensive than molecular tests, and the laboratory antigen tests are even less expensive than rapid antigen tests, around half the cost. Those of us, many of the people watching this, have purchased these rapid tests that range in cost from around $10 to around $15, $18. And the laboratory antigen tests are around $4 or $5, so they're even less expensive. The cons are that antigen tests may be somewhat less sensitive than molecular tests, but they're equally specific. And that may not be a problem. It might overcome the problem of persistent RNA in the absence of infectious virus that I spoke about. And the other big problem as we move through this pandemic is the results of at-home testing are not reported and not included in national data or local data. Some people may wonder, how can this nucleocapsid protein be detected in a sample if it's internal to the virus? And as I showed at the very, very beginning, when a virus infects a cell, it can actually lyse, releasing all of the building blocks, including nucleocapsid protein, from the lysed cell. And that material is present in an upper airway sample and detected by antigen testing. Now, detection of antigen can be done by these rapid at-home tests or by laboratory tests. And the rapid tests, there are 3 of them listed here, very commonly used, are available for purchase in many pharmacies unless they're sold out, which has been a problem in many parts of the country. These tests, basically, you take a sample yourself, you mix it with a buffer, and you place it in that sample chamber of the rapid test, shown by this person in this image. And typically what happens is that test card contains a line that we're talking about, the upper right panel, of C and T. The C is the control, which contains immobilized nucleocapsid protein that's immobilized on that strip of paper. The test line contains immobilized antibodies to nucleocapsid protein. And then the sample contains the patient sample, and it's mixed with a buffer solution that includes enzyme conjugated antibodies to nucleocapsid protein and some developing or color reagents. So when that sample is spotted in that well, it migrates upward, direction of reagent flow shown on the right. And if the patient sample does contain nucleocapsid protein, it binds to the antibodies on the T line that are immobilized on the paper, and then enzyme conjugated antibodies in the buffer will react with that and develop it as a line that gives color. And then also the enzyme conjugated antibodies do bind to the C line, to the immobilized nucleocapsid protein, just to show that the whole system is working. And on the bottom, the positive results can be either clearly positive on the bottom left, slightly positive, which means positive in the second image, negative in the third image, or invalid on the right side if the control doesn't light up. So many of us have used these, and they're fairly straightforward especially after you've done it a few times. It's actually, some people really have trouble using these rapid tests. There are high throughput assays offered by a number of companies, 3 are shown here, and these are somewhat superior to rapid tests in their performance. Some of the so-called laboratory-based antigen tests are actually low throughput, and they're just automated rapid test readers where you put the cassette in and it just reads that line rather than using the person's naked eye. But others are high throughput tests that can analyze hundreds of samples per day. So for example, the Diasorin and Ortho tests listed above, they can be walk-away tests requiring very little labor. The laboratory technologist just loads the samples, presses the button, and walks away. And again, the results can be available in a very short time, about an hour. How good are these tests? Well, there was a study that compared those rapid and automated antigen tests to RT-PCR for diagnosis of infection. And as you can see on the lower left table, for samples with CTs of less than 25 or less than 23, the sensitivity is quite high in the 90% range. Once you get to the 30s, CTs in the 30s, where the viral load is a little lower, then the sensitivity drops somewhat. And on the right side, if you compare the RT-PCR test with the laboratory antigen test and rapid antigen test on the bottom, you'll see that 50 of the samples, of these 87 samples, were actually detected by all three assays. And there were 5 samples detected by the laboratory test and the RT-PCR that were not detected by the rapid antigen test. How good are they also? Remember, using that RT-PCR as a gold standard, both tests perform with CTs of 25 or less. So people who are actively shedding virus are going to be positive with these assays. There is some thought that samples with CT above 30 may reflect that virus or free viral RNA below levels that are infectious. So for population screening, a high throughput test is very likely to identify infectious people very fast and lower than RT-PCR. The rapid tests have been used in large-scale studies recently published, showing that they're very good at detecting asymptomatic people. It's pretty amazing, actually, and that would probably be even more true of the laboratory-based tests. The pros of antibody detection are that serology can detect both ongoing and prior infection. It can be quantitative. Most companies offer high-quality serology tests, and there are rapid serology tests as well that can be performed at home. The cons are those rapid antibody tests are very expensive. Let's talk about diagnostics and challenges for laboratories and healthcare providers. Well, one of the biggest problems is that there's been a so-called great resignation during this pandemic, and clinical laboratories are really suffering as a result of that. Many people are leaving the workforce, including healthcare workers and laboratorians. And institutions can't afford to have the remaining workers out with COVID-19. So high throughput antigen screening has low labor requirements and allows the detection of infectious people without having the labor requirements of other testing modalities. So this is a real pressure on hospitals and clinical laboratories. Emerging trends. So we're going to see more and more of these at-home tests, and actually the US government will send a handful of tests to every household. Just need to go online and order them, and they arrive within about a week. The at-home antigen tests are already in widespread use, and this is likely to continue. However, many people have difficulty using these tests. And I have many friends who call me and say, oh, I just couldn't tell what it was, I couldn't get it to work, etc. These are educated people as well. There is COVID-19 fatigue. This, together with the low virulence of Omicron, will lead to continued relaxing of government mandates and individual measures that people can take to protect themselves. However, we need to remember this is still a serious illness for some people, especially the elderly, immunocompromised, unvaccinated, and those whose immunity has dropped over time. This virus will persist in the population through silent spread, even among vaccinated people, and even those who continue to wear masks and practice social distancing. It's very contagious. And since it's asymptomatic in many cases, people are unaware of being infected or of infecting others. And one way to identify those most likely to be shedding virus is through that antigen testing, which is the least expensive testing modality. COVID-19 in 2022. So the outlook. There is an institute called the Institute for Health Metrics and Evaluation at the University of Washington that publishes a briefing and is relied upon by many people in healthcare throughout the world as giving great forecasts for this pandemic. How accurate are the predictions? There's been look-back studies to see how the predictions were over time, and they were incredibly accurate and became more precise as time went on. There's a caveat to this, though, that I'll mention shortly. Currently, in the world, there are 24.3 million infections per day, estimated from last week, 10,000 deaths per day. 55% of the world population is estimated to have been infected at least once. 7% is the infection detection rate. That is, there are 14 times the actual number of infections than the reported infections. And Omicron remains the dominant variant throughout the world today. In the United States, we have 894,000 estimated and 166,000 reported daily infections last week. There are only 2,600 estimated and 2,100 reported deaths. You can see that the deaths and the estimated and reported deaths are closer to each other because it's hard to underreport deaths. Deaths are deaths and they're easily detected, whereas infections aren't. So there's a big disparity between 166 and 894,000. This makes COVID-19 the #1 cause of death in the U.S. Last week. 75% is an estimate for the percentage of people that have been infected at least once. 75%. And 9% is the infection detection rate in the US. So there are 14 times the number of actual infections than those being reported. Omicron dominates in the US as well. The 75% of Americans are immune to Omicron, either from vaccination or from prior infection. So that's the good news. In terms of the outlook for next year, we have a cumulative estimate of about 6.8 million deaths in the world as of June 1st of this coming year. 700 deaths by daily deaths by June 1st. So the number of deaths will drop to a very low level globally. 16 million cumulative deaths by June 1st. In the United States, by mid-April, we should only have less than 20,000 deaths per day, 20,000 cases per day, very low hospital census, and less than 1 million cumulative reported deaths by June 1st. Very low daily reported deaths by early April, and only a little bit over 1 million, 1.1 million cumulative total deaths due to COVID-19 by June 1st. Now, reinfections are becoming of increasing importance. And I would just emphasize here that a study in England that looked at 650,000 people, determined that more than 650,000 in England have been infected twice. Most were reinfected in the past 2 months due to Omicron. And before mid-November, reinfections accounted for 1% of cases. Now that rate is about 10%. So this is something we need to keep our eye on here as well. This silent spread is a major concern. And this became publicly obvious when many sports leagues, golf, NBA basketball, the NFL, and Major League Baseball, had lots of players testing positive who were largely vaccinated, largely showing social distancing and masking, but still getting infected. And this reflects this so-called silent spread. And there was one excellent study on this matter that was a longitudinal study in 2021. It followed 160 symptomatic people and 160 asymptomatic people, and all of their contacts were followed for 3 months by serial PCR testing. 87.4% of the positive contact cases were asymptomatic. That is, all those symptomatic people, only about 10% had symptoms. All those people who became infected. As expected, the PCR-CT values were higher in asymptomatic compared to symptomatic index cases. But there were no differences between the infectivity rates of index cases. So regardless of whether you have high or low virus, you were still very well able to infect others. This just demonstrates this problem. What are the challenges ahead? And how accurate were the predictions again? I mentioned how accurate they were, but what that IHME did not predict was the Omicron, shown in the blue question mark, this peak that just peaked at January and is now declining. So even though they predict no new spikes in the summer, April and beyond, that red question mark shows that we really don't know what's going to happen at that time. Emerging testing trends. At-home tests will continue to be in widespread use, and people will get more comfortable using them over time. Of course, due to this COVID fatigue, many people will not test at all, even if they have COVID-19 symptoms. Healthcare organizations and others, maybe sports leagues or other companies that need or want to reduce COVID-19, will implement regular screening of employees or members. Will be done mostly by RT-PCR, but also transitioning to antigen testing as a lower cost and equally useful alternative. My final remark. This SARS-CoV-2 is a perfect virus. It's very contagious. It causes few, if any, symptoms in most people. Vaccines work well for reducing spread, symptoms, and severe disease, but they don't prevent spread or reinfection. Silent spread will allow this virus to persist in the population for a long time, and animals that can harbor virus will contribute to this persistence, domestic animals and others. Public health officials will continue surveillance testing via RT-PCR and possibly high-throughput antigen testing. We just need to monitor this infection. Even if the hospitalizations and deaths go down, as long as that virus persists, it will continue to be a threat. And only time will tell whether a new variant will spike. And finally, waning immunity will lead more people to develop symptoms or require hospitalization and perhaps more boosters. So I hope that you enjoyed this presentation, and we'll have to see what happens in 2022 as we move through the pandemic onto a new phase of this disease, perhaps endemicity. And again, it's a major unknown what the future holds. But we have the armamentarium through novel drugs, vaccines, and testing to meet that challenge. Thank you very much.
Thank you, Dr. Engman, for that informative presentation. We will now start the live Q&A portion of our webinar. Now, a reminder to our audience, if you have any questions you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have so many questions that have come in. Dr. Engman, let's start with this one. How do you foresee testing needs changing as COVID-19 becomes endemic?
Wow, so I spent a certain amount of time discussing that, but I think one thing is pretty clear, and this COVID-19 fatigue is a real thing. Most people without symptoms, they'll never be tested unless they have a reason to be. The only reason that asymptomatic people would be tested is if they work in a field that tests, like healthcare, or if there's free, easy testing in the community where you go into the grocery store and you just test as a routine matter, or if they're included in a surveillance population. And that's one area that I think we need to do better in this country, at least, is to actually do routine surveillance to really understand what the level of this silent spread is and the persistence of virus over time. Serology can be important to assess immunity. I think we'll see an increase in serology for surveillance. And then I think we'll see institutions where an outbreak would be costly will continue to test. So, I think that what we're going to see is rapid tests on site with samples collected by trained individuals. I think that's the other issue, is that some individual people in the public, lay people, don't really know how to collect the sample. Those of us who've been sampled by healthcare workers know how uncomfortable it can be to get a really good nasopharyngeal specimen. Or maybe this high throughput testing on site or in a reference lab as well.
Thank you, Dr. Engman. Our next question. What has been your biggest surprise with diagnosing asymptomatic COVID-19 patients?
I think one is the large number, how large the number of asymptomatic individuals is. I was very surprised over time to know that it's larger and larger and larger. Up now it could be 9 out of 10 people without symptoms. The other issue is that asymptomatic people are possibly as likely to transmit virus as symptomatic patients. And we saw that from that study from Bahrain that I mentioned a little bit ago. I'm not terribly surprised that positivity on the antigen test correlates with the cycle threshold on molecular testing. That makes sense that the molecular testing is quite quantitative in terms of the viral load. I am surprised that RNA can be positive after intact virus has been cleared. And the evidence for that, by the way, is that those samples are introduced into tissue culture, and evidence of live virus can be seen in those cultures, and that you can have samples that are RT-PCR positive that do not contain active virus. And I think that finally, I'm a little surprised that serology has not been implemented on a more routine basis. But I think that's coming, especially for healthcare workers, where you sort of want to know your healthcare workforce, how many people have been infected, vaccinated, monitoring vaccine efficacy and antibody levels, et cetera. And possibly we might even see some T-cell assays developed that might correlate better with protection from future infection.
Dr. Engman, what lessons learned can we leverage from COVID-19 to improve our lab testing moving forward, maybe for the next pandemic?
That's an excellent question. I think one of the things that first of all, the testing industry globally and in academic labs and in corporations has been really amazing to watch over the last several years. It's been outstanding. I think we could have done even better if we'd established a set of standardized control samples that could be distributed to anyone who wants to develop a test, whether it's a company or an academic lab, for a diagnostic test development. This way, we're comparing apples to apples, and especially when people publish those early studies about sensitivity and specificity, they're using different sets of samples. Often they're from high titer viruses where it's very easy to detect a positive sample and get a sensitivity rate that's much higher than you might get from an asymptomatic or from somebody with waning viral load. And I think that there have been a lot of work recently to establish in proposals a pan-pathogen metagenomic approach. This is where you basically can use RNA sequencing to a sample, to characterize patient samples without any knowledge of what the type of pathogen could be, viral, bacterial, parasitic, fungal, and also pan-viral family PCRs to investigate an outbreak for early pathogen identification. So once you identify this, then you can use a specific PCR or antibody or antigen test. And you don't even need to have isolated organism. All you need is the sequence, and you can produce all of those reagents for diagnostic purposes and surveillance.
Thank you. Our next question. The overall case fatality rate that you mentioned, does this include vaccinated and unvaccinated?
It does. The overall rate does, but it's very clear that deaths are much more likely to occur in unvaccinated people. I haven't recently seen the deaths broken out by vaccinated versus unvaccinated, but I'm pretty sure those numbers are available somewhere. We'd have to look.
Now, Dr. Engman, what kind of benefit do you see from doing the SARS-CoV-2 variant surveillance, since Omicron is being the major variant with mild symptoms out there now?
I think at the present time, it's the current level of sequencing that's happening globally. And that GIS-AID resource, where people are submitting sequences from all over the world, has been pretty remarkable at tracking viral variant emergence. And that will continue unless people decide to drop that, which I think it's really premature. I mean, I think if you look at the Omicron case fatality rate and the virulence, it's quite low. And I think that's, especially as we move into our own summer season here, spring and summer, people are just not going to tolerate some of the mitigation measures. And I think that if we drop our vigilance on that global sequencing and sampling effort, we might be caught in a bad place if a new variant does emerge in the population. There is already, I didn't point this out too much, but in that chart of the United States, that BA2 variant, which is a subset of the Omicron, is starting to emerge and increase in frequency compared to the previous, the major Omicron variant that dominates at the present time. And so that could then emerge. But it might be a non-Omicron variant that pops up later, but nobody knows for sure whether there even will be another variant that emerges to dominate the global scene like Omicron has, and Delta before it.
Thank you, Dr. Engman. Now, this question is two parts. First part, do antibody levels correlate with protection from COVID-19? And couldn't these levels simply be measured and boosters given to people whose antibodies fall below a certain level?
You know, I think that when people first developed these serology tests, that was the hope. The hope is, and actually anyone here who works in a healthcare organization that requires titers for a variety of childhood vaccinations be measured and then boosters given for measles or mumps, rubella, whatever it may be, knows that they're basing that on the result of a serology test that's done as an adult well past that vaccination time. I think the thought was that maybe the same thing might be true of SARS-CoV-2, that you could just measure antibodies, and once they fall to a certain level, that then the person's not protected anymore. But the correlation between antibody titers and infectability is not straightforward, and there are people with quite high titer antibodies who get reinfected and sick, and people with low titer antibodies who, as a population, do not get ill. So that's been a little disappointing for people who wanted an easy biomarker for, protection from infection. There's been a lot of work on trying to identify other markers that might be more useful that way, and there's some promising candidates, but nothing that is emerging as the gold standard test to see whether some vaccinated or infected recovered person is protected from reinfection. And what was the second part of the question? Or maybe I did.
Yes, the second part was, sorry, let me roll back to the picture. Couldn't these levels simply be measured and boosters given to the people whose antibodies fall below a certain level?
Okay, yeah, I incorporated that into my question. That it was the hope, but I think that is not turning out to be as useful as people hoped.
Right. Thank you for that. All right, our next question, and we have so many questions coming in. I do want to remind our audience that any questions that we are unable to answer today, they will be answered via email at the email address you used for your registration. Okay, next question. Now, Dr. Engman, there appears to be little specific information regarding the impact and the implications for the transplant and oncology communities. With Omicron's high level of transmissibility, are these populations essentially, quote unquote, unvaccinated despite having had 4 vaccinations?
So there was great concern, and justifiably so, about people who are immunocompromised for a variety of reasons, transplant patients, cancer patients, etc., because of their potential increased susceptibility to this infection. And so there have been studies well before we even started talking about boosters, where some of those populations were given 3rd doses, 4th doses of vaccines, and those actually have been reasonably successful at boosting antibodies up to reasonable levels, whereas people after 1 dose or 2 doses who are transplant recipients, for example, made very little antibody and were highly susceptible then to infection, presumably susceptible. So I wouldn't say that the solution to protecting immunocompromised patients is simply more and more boosters, but it's made some theoretical headroad into solving that problem. I think if I were an immunocompromised person, I would be vaccinated as many times as allowed and practice social distancing and masking and mitigation measures.
Thank you, Dr. Engman. And we have time for one final question. Given how the lab workforce has been impacted by COVID, do you see testing changing to accommodate the staff shortages?
So this great resignation, as it's called, it's not clear that that's going to end with a resolution of this pandemic if it resolves. I think a lot of people have used these last few years to really look at their own personal lives, their own financial situations, etc., and making decisions either to change jobs to do something where they can work from home, which obviously isn't viable for a laboratorian, or not re-enter the workforce. So that's one thing. And I also think that the impact on lab staffing depends on the lab. So some labs have been able to manage these surges in testing by shifting around personnel, adding extra shifts, but others have either outsourced or even closed temporarily because they just couldn't handle those loads. And I foresee this variability in how individual labs handle these surges continuing, but many labs have implemented cross-training of people in other parts of the laboratory to be able to handle those increases that might be needed but temporary.
Dr. Engman, I'm going to squeeze in one final question here.
Okay.
Could you just comment a little bit more on the infectious profile based on the cycle threshold view?
Yes, I think that there are a number of studies. I pointed out this one from Bahrain that showed those 90% silent transmission among 160 symptomatic and asymptomatic index cases. What was found there, though, is that there's no difference in the infectivity rates of the index cases depending on the high or low CT. So people who have very high CT values, low virus, are just as infective as people with low. Although, if you look at the index cases, those who had symptoms had much higher virus, lower CT, than those without symptoms. The asymptomatic people are likely to have high CT or low virus. So that was very interesting, is that symptoms in the index cases correlated with CT, but not in the infectivity of the cases that arose from contacts of those index cases. So it's very interesting.
Dr. Engman, thank you so much for your presentation and for your insightful answers to all of our questions. Do you have any final comments for our audience today?
I think that, first of all, thank you everybody for tuning in and for your questions. I've watched some of them. I should have been speaking, but I come in and they're very interesting. There are many that we were not able to answer. I think that we all should be optimistic, but also very vigilant as we move through 2022. This is not over now, and there's no guarantee it's going to completely end here. So I think all of us need to work together, and hopefully we will emerge from this better in our ability to diagnose and manage pandemics, and smarter about how we understand viral pathobiology and immunity and vaccination.
Thank you again, Dr. Engman, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thorson, for underwriting today's educational webcast. Before we go, I do want to thank our audience for joining us today and for their interesting questions. We had quite a few questions we weren't able to answer due to time, so those questions that we did not answer today and those that are submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us, and have a great day.